Hello again, beautiful biology students. We are going to wrap up Chapter 13, Lesson 3 on DNA replication. In this lesson, we're going to learn how it is that DNA unwinds to create two identical DNA molecules. Now, this process is going to be full of many actors to include helicase, DNA polymerase, and many more. So let's get started. What does replication even mean? Replication simply means to copy or reproduce. In this case, we want to reproduce the DNA molecule. Why does DNA replicate? Remember, every time our daughter cells divide, we want to make sure that each daughter cell gets equal amounts of genetic information. So, when does DNA replicate? DNA must replicate before the cell divides. In eukaryotic cells, this will occur during the S phase or synthesis phase of interphase, before mitosis or meiosis. In prokaryotic cells, the DNA will replicate in binary fission before the cell divides. Now, before we begin all the steps, we need to remember that DNA is antiparallel, and when we start to add new DNA nucleotides to the DNA strand, they can only be added in a 5' to 3' direction. Here is a model of our DNA, like we built in class. And here you can see that the DNA double helix is slowly unwinding. How is that happening? By breaking those hydrogen bonds between the base pairs. The place where the DNA double helix starts to unwind is called the point of origin. And it will form replication forks on either side, one here and one over here. The DNA double helix will continue unwinding in both directions. Now, it's important to note that in eukaryotic cells, we have many replication forks because we have many points of origin, even though there's only one being shown here. In prokaryotic cells, because the ring of DNA is so small, they're only going to have one point of origin. So here we have a drawing of the DNA molecule that has been unzipped. We have our point of origin showing and our two replication forks forming. So who is responsible for breaking these hydrogen bonds that hold the base pairs together? That's the job of helicase. Now, there will be several helicase enzymes binding to the DNA molecule in eukaryotic cells. Helicase will move along the DNA strand, breaking the hydrogen bonds as it goes. It might be useful to memorize H for helicase and H for hydrogen bonds. Now, helicase has some special helpers as well. There are single-stranded binding proteins that will also bind to the DNA molecule, and their job is to keep the DNA molecule from re-zipping together. Before we bring up the next enzyme, let's remember that DNA is antiparallel. We will put the 3' end here, the 5' end here, 3' end here, and 5' end here. Now, at our point of origin, we said that DNA nucleotides can only be added in a 5' to 3' direction. I'm going to focus right now on this part of our DNA molecule. This will be called a leading strand. There is also a leading strand on the other side of the DNA molecule over here. Again, notice we will be adding nucleotides in a 5' to 3' direction. And again, we will term these the leading strands. From the point of origin, our enzyme primase will begin adding RNA nucleotides to the DNA template strand in a 5' to 3' direction. Here I'll show primase adding new RNA nucleotides to the DNA strand in red here in a 5' to 3' direction. This again is called a primer, and it's made up of RNA nucleotides. Our next enzyme to show up is DNA polymerase. DNA polymerase is the builder. This enzyme job is to add new DNA nucleotides to the original DNA strand. Again, just like primase, DNA polymerase can only work in a 5' to 3' direction. So, how does DNA polymerase do it? Well, it's going to add new nucleotides that have complementary bases to the original DNA strand. Notice again that DNA polymerase is working in a 5' to 3' direction. And it almost appears like DNA polymerase is chasing after helicase. And the reason why this is called then the leading strand is because DNA polymerase can work continuously as the double helix is unwinding. The next enzyme in our process here is ligase, and ligase's job is to glue the new nucleotides together. I like to think of L for ligase, L for link. And I'll show it here in yellow, these phosphodiester bonds that ligase is forming between the new DNA nucleotides. Now, we don't want RNA nucleotides left in our DNA molecule, so a different type of DNA polymerase will come along, and it will actually remove the RNA primer, and it will replace it with complementary DNA nucleotides. And so again, this is called the leading strand because DNA polymerase can work continuously in the same direction as helicase is traveling, that 5' to 3' direction. Now let's talk about the lagging strand on the other side of our DNA molecule. Why is this called the lagging strand? Well, it's almost like our enzymes have to work in a backward direction. So let's see how it looks. Once again, primase comes in first, and from the point of origin, primase wants to add a primer made up of RNA nucleotides. But remember that primase can only work in a 5' to 3' direction. So from the point of origin, we'll begin adding our RNA nucleotides to the original DNA strand. Now, we're still working in a 5' to 3' direction. It just looks as though they're working backward from that replication fork and the direction that helicase over here is traveling. DNA polymerase comes next. DNA polymerase will add new DNA nucleotides that are complementary to the original DNA strands, using those hydrogen bonds between the bases. And what we're forming here is called an Okazaki fragment. Okazaki fragments are these short sections of DNA that are being placed by DNA polymerase in the 5' to 3' direction. But notice that the sections are laid moving away from the replication fork and away from the direction that helicase is traveling. Ligase comes in next, and again, ligase's job is to create those phosphodiester bonds between the DNA nucleotides. And also don't forget that we want to get rid of this RNA primer, and that will be done by a different DNA polymerase enzyme. This enzyme will add, of course, DNA nucleotides to the original DNA strand. So just a quick review here. We have the point of origin where the DNA helix starts to unzip. The replication forks form on either side of the point of origin. DNA is anti-parallel. New DNA nucleotides can only be added in a 5' to 3' direction. On the side of the DNA strand where DNA nucleotides can be added continuously in a 5' to 3' direction, these are termed the leading strands. When DNA nucleotides have to be added in short sections moving away from the replication forks, these are called Okazaki fragments on the lagging strand. The end product of DNA replication is two identical DNA molecules. Notice that each DNA molecule is composed of half ladder that is old, or the original DNA strand, and a half ladder of new nucleotides. It is for this reason that DNA replication is referred to as semi-conservative, since the end product are two DNA molecules where each DNA molecule is composed of old and new nucleotides. So let's play a little review game here. You can pause the video to give yourself time to guess. So, which enzyme's job was it to break the hydrogen bonds holding the base pairs together in the original DNA molecule? That's correct, helicase. Now, which enzyme's job was it to lay a primer down made up of RNA nucleotides? Correct again, primase. Which enzyme has the job of adding complementary DNA nucleotides to the unzipped DNA strand? Correct, DNA polymerase. And which enzyme's job is it to link the new DNA nucleotides together to create that sugar phosphate backbone using phosphodiester bonds? Correct, ligase. Great job, beautiful people. We're getting ready for that big test coming up. Good luck, everybody.